everybody to this inaugural professorial lecture. When we think of the school, when we think about what the school means to us, and the most important part of the school, it obviously is our staff. And it's always good to be able to recognise our excellent staff, and particularly those staff who have been promoted within the university. And so it's a great pleasure to be able to welcome you all here to Iain Lamont's inaugural lecture. And it's also a pleasure to welcome Iain's family here to come and join us for this very special occasion. And so without further ado, I'd like to introduce Professor Kurt Kraus, Head of Department of Biochemistry, who is going to introduce Iain to you.
It's my great pleasure, of course, to speak and introduce Iain on the occasion of his inaugural professorial lectureship. I thought I would start out and just say a couple words about his academic history, a little bit about the kind of research he does, and a little bit about some distinctions. Iain received a BSc Honours in 1980 from the University of Edinburgh, and then following that he matriculated at University of Oxford, and he received his DPhil at Oxford in 1983. After finishing at Oxford in 1983, he received a National Research Fellowship that he took up at the University of Adelaide, and he was at the University of Adelaide for the next 3 years. He moved then in 1987 to join the Department of Biochemistry at the University of Otago as a lecturer, and then he worked his way steadily up the ranks until in 2003 when he became an associate professor. And then this year he received the rank of professorship. Iain's main interest has been in infectious diseases, and his work has focused predominantly, although not exclusively, on Pseudomonas, which is a very, very important cause of disease. And it's a tenacious, ubiquitous, difficult to eradicate organism that I know from my clinical work is one of the toughest challenges that we face in clinical medicine. And he's focused on some specific aspects of this organism that he'll share with us tonight, but most specifically its use of iron, its development to sense and acquire iron, and it's a signaling mechanism by which it's able to signal the presence of iron and mount an official mechanism to claim iron from the environment. Iain's had a number of distinctions in his career, which I'll list just a few. He's the New Zealand Microbiological Society Distinguished Speaker this year at an upcoming meeting. He's received the New Zealand Society of Biochemistry and Molecular Biology Award for Research Excellence. He was a Commonwealth Medical Fellow. He's a past president of the New Zealand Microbiological Society. He's been active in a number of service positions at the university. For example, at the University of Otago, he's been the chair of the IBSC since 2003, and he's the newly appointed successor to take over the genetics teaching program beginning next year. I think Iain, to me, has been an inspiring scientist and a compelling teacher and a wonderful colleague, so it's my great pleasure to introduce him to speak about his research tonight. Iain?
Okay, well, thank you very much, Kurt and Helen, for the kind words in the introduction, and thank you everyone for coming along. And I'm looking forward to telling you a bit about what we do, if this will let me. But also, I'd like to take the opportunity to tell you a little bit about why we work in this particular subject. I think in all the time that I've been at Otago, nobody has ever asked me why do we work and what we do. So I thought I'd tell you. So hence the title of the talk, Bacteria are an infection, what do we do and why do we do it? And the why do we do it really stems from a sort of series of historical events. So I'll probably go through that first of all and then come back to the what do we do as things move on. Okay, so first of all you can say, why do biochemistry? There's lots of interesting things to do with life. Why would I study biochemistry? And it really stems from when I went to high school. And Kurt didn't mention, but I went to high school in St Andrews in Scotland. Many years ago now, but I don't think the school have changed very much. This is our school, which was very old when I was there, and even older now. The actual classrooms behind, this is the administration block. In front of the school is a big lawn. This is a view looking the other way, where you see this big old building. It's not actually part of the school, but this is actually a monastery it was built in about 1100. By the time we got there, it's a ruin, but with hindsight, it's quite cool having a 900-year-old building in front of your school. I don't think there's many like that in New Zealand, but it's got nothing to do with biochemistry, so I better move on. So, what I like at school, I like chemistry, and the computer has converted my tick into a square root, but chemistry was good. I like biology, so I enjoyed those all the time at school. And in the last year, I suddenly discovered that chemistry and biology could connect. So our biology teacher got a bit adventurous and told us about this wonderful thing called biochemistry, and he told me that chemical reactions can actually happen inside cells. And there's just one example of the kind of thing that we heard a little bit about, and for the biochemists in the audience, you'll recognize this as the first step in the breakdown of glucose, which is a chemical molecule that we use for obtaining energy. So I was fascinated. I was hooked by the idea you can think about living things in chemical terms. So inspired by that, I went off to university, and there I discovered bacteria. And why bacteria all come to university first of all. So as Kurt mentioned, I went to university in Edinburgh. This is not the university, this is the old town in Edinburgh. I put this picture up partly because this house is a bit of a historical landmark, but also because I actually lived 2 buildings down the road in that place there. And that's actually really cool, being right in the middle of Edinburgh. I appreciate it at the time, and with hindsight, I appreciate it even more. But that wasn't where I spent most of my time. I spent most of my time in this building here, which is actually where the University of Edinburgh had its microbiology department, which is about 20 minutes away from the centre of town. And this isn't quite so nice photograph, but I think brighter sunlight would not make the building look much better. However, I discovered bacteria there, and bacteria I thought were great because they're what at the time seemed simple organisms. Unlike us, would contain hundreds of millions of cells. Bacteria they're single cell organs. One cell does everything. And that I thought made them nice and simple, and probably shows how simple I was at the time, but that made them simpler things to study. So this is bacteria. This is a single cell looking down an electron microscope, a very powerful microscope. This is actually bacteria grown on a petri dish, which many of the scientists in the audience will be familiar with. This is a dish roughly the size of a compact disc, and you cover that with a gel that provides all the food the bacteria need to grow. And one of these cells has been put on the dish at each of these points and has grown up and made lots and lots of new bacteria. Each of these little sort of pinhead-sized things will contain hundreds of thousands, perhaps millions of bacteria. The other thing I discovered at Edinburgh was that bacteria aren't just biochemically wonderful, they're genetically wonderful. So the bacteria is quite straightforward to obtain. Mutant strains lack specific proteins, specific enzymes that do specific jobs inside the cell. And I was intrigued by these because they can help you understand biochemistry, which I was and am in love with, but also they're interesting in themselves. And what I forgot to do, I actually found in my office, that I'll never throw anything out, I actually have a copy of my honours dissertation from when I was at Edinburgh. I was going to bring it along as I had to somehow justify keeping it for 30 years, but I missed the opportunity, so another 30 years we'll see what I can do with it. Okay, so after Edinburgh I went to Oxford to carry out my PhD, and there I worked on this bacterium called Bacillus subtilis. And these, like the one I showed before, are really cylindrical things, but they're quite unusual, I guess, amongst bacteria, that they can grow not just in this form, but they can go into a completely different form, which is shown here. And this is a spore, which is a resting stage, which is very resistant to desiccation and heat. And these bacteria make these, and then they can live in the soil dormant for a long, long time. And they make them when times are bad, and then when times are good, when food comes along, they can reverse the process. So I was intrigued by this. I learned about it a little bit in my undergraduate. And then the leading person in Britain working in this field was this gentleman, Joel Mandelstam, who was working at University of Oxford. And I contacted him about the possibility of doing a PhD and ended up going there. And Joel was a great guy. He's a really inspiring supervisor. He's very bright, very smart guy, but he also ran a very nice group, and he's a lovely person. So I had great fun doing my PhD. This is actually a picture from my PhD thesis. And this shows that as the bacteria transition from going from this growing form to this resting form, they go through a number of defined stages. So what I was doing in my PhD was essentially obtaining mutants of the bacteria that had lost the ability to go through this whole process. And it turned out that they were blocked at different stages. They might get this far and then get stuck. They might get this far and then get stuck, or whatever. And we used these to try and understand the genetics of what was going on. And basically what is known now is these bacteria contain probably 50, perhaps a bit more genes that are required for this whole process to take place. So that was really, I guess, classical bacterial genetics, taking cells, getting mutants that have lost the property to do something. And around about this time, in the early 1980s, was just starting to become widespread with what we now know as molecular genetics, where you can actually study genes at the molecular level. So what we could do here is you get bacteria and say, okay, they got stuck here, that's because of a mutation in a gene, but we had no idea what that gene was actually doing, why that mutation in that gene had that particular effect. So what we want to do is to do that. And as I said, this discipline of molecular genetics was just really starting to fully flower. So molecular genetics, I've used the description of how genes work. So I've already talked about genes, they're the units inside cells that essentially give the cells their property, but the question is, how do they actually do that? So in the 1980s, methods for actually purifying genes out of cells were just becoming commonplace. Before that, all you could do is to say this cell contains 5, 6, 7,000 genes, but there's really no good way of separating one from another. Okay, and the younger people in the audience probably can't even envisage what that was like, but for us at the time, that was a big challenge. So I was really intrigued in how you can do this, because then you can start analysing single genes in detail to see how they actually work. So I looked around at a number of possibilities, but what I ended up doing was going to work in Australia at the University of Adelaide with this guy called Barry Egan. And Adelaide was really at that time the centre by far, the best place in Australia for looking at this kind of molecular genetic research. So that's a really good place for me to go. What Barry was working on was not bacteria itself, but a virus that infects the bacteria. And when I said bacteria are really small, these things are really, really small. So this says 100 nanometers, so the bacteria I didn't talk about, but this says that you can get 1,000 of these, and if you stack them up, you get something that's a millimeter in size. So in biological terms, that's pretty tiny. So why is Barry working on this, and why was I interested in it? Well, really it could do quite a lot of things that were a lot of fun. Oops. So first thing is, even though this is very simple, it only contains about 40 genes, it's got two alternative lifestyles. Like the bacteria and the bacillus that I talked about before, you can switch between two different forms. Now you'd think a virus couldn't really do that. What this virus does most of the time for its, I guess, main lifestyle is it'll infect bacterial cells, make 40 or 50 copies of itself, kill the cell, and then the virus gets out and can go off and look for no more cells to live on. It has an alternative lifestyle where it'll infect the bacterial cell and then nothing happens. It sort of vanishes. The bacteria just keep on living. They're quite happy. What the virus has done is that it hasn't completely disappeared. It's taken its genetic material that's stuck in this structure here and incorporated that into the DNA, into the genetic material of the bacterial cell. So it's gone into a dormancy mode. And that can go on for a long, long time, and then every so often, for no reason, the virus will go back into its killer mode and the bacteria and make more copies of itself, and the bacterial cells will die. Okay, so I was intrigued by that aspect. The other thing was that this is really great for doing molecular genetics because it's quite small. It only has 40 or 50 genes, which means it also has a relatively small amount of DNA, so purifying 1 or 2 genes out of that is quite straightforward. There are also good methods of doing traditional genetics of the kind that I talked about before, and those kind of go hand in hand. The other thing that became clear to me while I was working in Adelaide on that system is what is crucial is not just which genes you have, but which ones are active at any one time. And the virus is a really good example of that in its dormancy mode, only probably one, maybe two of its genes are working, the rest are completely silent. In its life mode, basically every gene is fully switched on and is making more and more viruses. So, as I put in the words of the song, what you got is the way to use it. It's the control of gene activity when you make them work, as opposed to when they're not working, that really tells you, that really determines what, in this case, a virus, but also how a cell is going to be working. Okay, so as Kurt said, in 1987 I came to Otago. Things were a bit more informal than when we appoint people nowadays. Nowadays we have a very complex selection process where people will apply for a vacancy. We obtain referees' reports from some of the candidates. We'll send some of them here, have some of them come here for interviews. We'll give talks about what they do. We'll discuss their research. We'll discuss their teaching. It's really a very detailed process. In those days, it was much more informal. So the head of the department at that time was George Peterson. What happened was that basically I wrote a letter to the department in response to an advert they put out saying, I'd like to apply for this position. I heard nothing more about it for a while. Then when I was in the lab one day, just to finish off an experiment, I got a phone call from the dean where we had half an hour discussion about what I'd teach if I came to Otago. Then I heard nothing more about it until I had a letter in the mail telling me that the job was mine. Okay, so I thought that was good. I knew nothing about Otago at all, except that I had a friend from Oxford who was here who told me that the department was a good place to work. He's absolutely right, it's been a fantastic place to work. But I got here and there'd been a big focus, not a big focus, the entire focus of our conversation had been what we would teach on. Nobody had any discussion about what research I would do when I got here. And after I arrived, just the same was true. Turned out when I got here, about 4 members of the staff were on leave, so I sort of thrown into teaching. I sort of got here on a Sunday, and about the next Thursday I was running my first class, and the week after that I was giving a session to the fourth year students. And that was great fun, but beyond that I sat in my office and thought about things, and I was a bit surprised nobody came and said, so what are you going to get up to then? But I figured that I should do some research. So I said, well, what will I work on? And it was a wonderful opportunity, once in a lifetime opportunity really, to decide what's going to be the best thing to work on. So what would I like to work on? And this is really coming back to why do we work on what we do. So I really scratched my head and thought about this quite a lot. And I wanted something that was going to fit in with what I'd learned already, and with my interests in working with microbes, but also with bacteria, with biochemistry and with genetics. So I wanted something that's going to be genetically characterized, not an organism that nobody knew nothing about, something that was tractable, in other words, you could manipulate it reasonably easily, genetically. Something that would be suitable for students to work on, so not something that was going to be really hard or really dangerous. Something to be suitable for carrying out biochemistry. Some of my thoughts would be relevant in medical school, but yeah, well, you can see that bit yourself. I thought it would not look good if my first student died of bubonic plague. So these are my sort of criteria. I looked around in terms of what I worked on already and came up with the magic answer, which is Pseudomonas aeruginosa. And many of you will know there's a bacteria that we worked on pretty much since I started, which we still handle very intensively. So this basically ticked all the boxes. It's fast growing, it's easy to handle in the lab. People already had done these classical genetics with it, so we knew a bit about the genes for it. It doesn't infect healthy people, but it infects people who've got various pre-existing conditions. So the examples I've given are cystic fibrosis or severe burns and implants, etc. And this suited me very well because I thought it was unlikely I'd have students in the lab with cystic fibrosis or severe burns or implants, so they'd probably be safe even if they didn't handle the bacteria very well. The other thing we knew already about it was it secretes various toxins and enzymes during infections, and this was of interest to me biochemically. So these are the bacteria. There's actually a picture I showed you before. These are pictures of the bacteria that, like the other ones I showed, are sort of rod cylindrical structures. This is actually a diagram representing that in a reasonably cartoony form. So what you have is a sort of envelope which surrounds the bacteria, which basically keeps everything inside. In the middle of that, you have the DNA, the genetic material of the bacteria. And then the envelope is actually quite a complex structure in this species. So it's got two different membranes, all cells are surrounded by a single membrane which keeps everything in and controls what goes in and out. In bacteria, in these bacteria, you have a second membrane that essentially adds to that control of trafficking of chemicals in and out of the cell. The cell wants to make sure that good things get in, food, nice stuff, but doesn't want to let in things that will be bad for it, and also needs to get rid of things that it doesn't need anymore. Okay, so if you remember right back to the start, then I put iron and infection into the title, and I've talked about those. How does iron fit in? Well, we know that we need iron in our diet, whether you get through spinach or eating red meat or buying iron-containing tablets. But bacteria need iron as well, and we and bacteria need iron because it's a crucial component of some enzymes that we actually absolutely have to have if we're going to be able to live. So bacteria need iron as well, but they don't have the same option as us. They're not particularly great at going down to the supermarket and buying a pack of vitamin tablets. So what do? The problem for the bacteria is that where they live, iron is not readily available. So Pseudomonas lives widespread in the soil, picture of soil, but in soil, the iron is very insoluble. It's really, it's like microscopic rocks. It's not just ready to diffuse into the bacterial cells. In people, you'd think, well, there must be iron in us because we eat iron, and that's right. But the iron is tightly bound up to proteins and not in a form that's just waiting to get inside bacterial cells. So the bacteria have a problem. And they get around this by secreting compounds that are really very good at binding iron that can go and hunt the iron for them so they can get it inside their cells. Okay, so I put up a few examples of these for if you're chemically orientated, then you can feast your eyes on these beautiful things for a moment or two. If you're not so chemically inclined, then watch my hands. So what the bacteria do is they secrete these chemicals out of themselves, and essentially they go around looking for iron molecules. When they find one, they latch onto it very, very firmly. And then they can take that up again and deliver the iron back into the cells. So that's how they get hold of the stuff. Time to come this side, I think. Okay, so that's what I've tried to represent in this little diagram. So this is meant to be a bacterial cell, and here are the two membranes on the outside that I talked about. And in the middle, there's all the DNA that contains all the genes that the bacteria need to be their happy little selves. And out here, there's an atom of iron that the bacteria wants on the inside because they need the iron to be able to grow. So what happens? So bacteria, the cells, just like ours, are chock-a-block with all the chemicals that they need to be alive. And I've represented these in a rather non-chemical form. And what the bacteria wanted to do is make one of these iron-chelating compounds. So what they have to do is to essentially assemble that from a whole lot of different bits. So these are the bits that they're going to assemble into one of the iron scavenging compounds that I talked about before. So how do they do that? What they have to do is to really, these guys aren't going to come together all by themselves. The bacteria have to make a whole set of enzymes that are going to assemble all the bits into the iron scavenging compound. So what happens is the genes in the bacteria make these enzymes and join them all together. Then the iron scavenger is made and secretes out of the cell, and it's going around looking for the iron that it can latch onto. When it finds some, joins onto it to make a complex, so the two join together. And that is on the outside of the bacterial cells. It's no use there. The bacteria want it inside. To get it inside, what they do is they have special proteins that sit on the outside of the cell, which are receptor proteins that very specifically bind these guys and take them inside. So these aren't able to get through the membranes all by themselves. They have to have a protein to help them out. So this binds to the protein. What happens is that the iron is transported inside and the chemical was sent out, the iron scavenging chemical was sent out to go and look for more iron. So this is how the bacteria get around the problem that the iron, the very little iron that is around, the very scarce iron, they can get inside the cells. So in outline, we knew that before I started Otago. In Pseudomonas, we didn't know anything about it, except that we knew that they made this kind of thing, but in principle, this had to be how things worked. So this is the molecule that we knew about at the start, Pseudomonas iron scavenging molecule, pyroverdin. And what I've tried to do in this talk, I've tried hard to try and keep technical terms to a minimum. But this is the only one that I think I'm going to use, pyroverdin. And there are two reasons for that. The first one is I'm probably constitutionally incapable of giving a talk without mentioning it, at least by accident. The second is that I don't think it should be just a technical term. This is a fantastic molecule. I think everybody should know this word. So I'm not going to rest until there's turning up in crosswords and in pub quizzes, and you guys know the answers, right? So pyroverdin. So it's actually a nice fluorescent molecule. So what are we showing here? These are plates of the bacteria, like I showed before. These bacteria here, in something resembling my name, are making the pyroverdin, which is this wonderful yellow-green fluorescent compound. These bacteria are not, so you can see the very clear difference. I should say that anybody who knows me will know I didn't make this plate. I can't write this well with a pen, never mind on a petri dish. So this was made by Lois Martin, who works in my lab. So these are the bacteria growing, instead of growing on a gel, growing in liquid culture. And these guys are making the pyroverdin, are nice and fluorescent, whereas these ones that are much more dull, they're not making the compound. And then there's the compound underneath. All right, so when I started, what do we actually know? Well, we knew that the pyroverdin was made, but we didn't know a lot about what controls it. We know if you add iron to the growth medium, then the bacteria don't make it anymore. That sort of makes sense. A, they want to save energy. B, if you have too much iron, if the bacteria take up too much iron, they're going to poison themselves. People can get iron overload syndrome, so can bacteria. So we knew that iron controls production of the pyroverdin, but also production of two different proteins the bacteria secrete when they're trying to cause an infection. But that was it. Okay, that's all we knew. At the mechanistic level, actually, how does that work? What's happening inside the cells? That had only been characterized in another species of bacteria, which has got some similarities to Pseudomonas, where people identified an on-off switch as a master controller that's determined inside the cells that when too much iron was around, then things would be shut down. So I'm going to tell you just a little bit about that, because that forms a sort of platform for what we did with the Pseudomonas. Okay, so this is again representing our bacterial cell. And here is one of these Escherichia coli cells busy making this compound and taking up iron and being happy. And let's see, it does a good job. So the key protein that I've called inhibitor protein, its job is to control the activity of these genes. When the cells contain lots of iron, they'll be quiet. When the cells contain very little iron, then these will be active and the iron scavenging compound gets made. How does that happen? How can a protein do that? Well, the answer is that this protein is only really active when it joins onto iron. So if the cells don't have much iron inside, then all it does is sort of swim around waiting for a good day. If the amount of iron inside the cell builds up, that joins onto the protein, and that then latches onto the DNA, onto the genes that are making these enzymes. When that happens, then now the genes can't work anymore. Okay, and if they don't work, then they don't make the enzymes that are needed for joining the jigsaw pieces together. And if you don't join the jigsaw pieces together, then you don't get your chemical being made, and then everything goes quiet. Okay, so that was a molecular switch that was present in this species, Escherichia coli. What actually happens in Pseudomonas? So these are the questions that we had. How is iron scavenging controlled in our species as opposed to this other E. coli species? And also, how does this relate to the production of the toxin and the protease that I talked about before? I told you that these that are important in infection are made by the bacteria unless you add iron and when they stop making it. So, does production of this connect to production of this? All right, so my long think after I arrived in Dunedin was along the lines of, this is the puzzle, how do we actually solve it? And this is the approach that we took. we said, all right, let's purify the genes that are needed for making this chemical, the pyroverdin. Once we purified those, we can analyze those. We can also use them to determine the mechanisms that control their activities, what determines when they're on, what determines when they're off. And last of all, once we've identified that for pyroverdin, the iron scavenging compound, let's see if the same mechanisms control production of these guys. Again, older members of the audience will remember a time when polymerase chain reaction didn't exist. Polymerase chain reaction, essentially a compulsory tool in all biochemistry type labs these days. And it's hard to imagine a time when it didn't exist, but once upon a time it didn't. And a long time ago, when I started, that was the case. So what I'd learned in Adelaide was how to actually purify genes by doing this method called cloning, where essentially you're purifying the genes out of the bacteria. So we did this, and when I say we did this, what I mean is that about 3 different PhD students worked on this. So it took quite a lot of time, but we purified 3 different genes, and we had proved that they were actually involved in the process that we thought we were by engineering the Pseudomonas bacteria so that those genes were inactivated, and then the bacteria couldn't make the enzymes, and then the process of making the compound stopped. So this is a plate I showed before. This is our bacteria that are making the compound. These were one of the strains that we engineered that have lost that ability. So that proved we'd actually identified the genes that we wanted. So then we said, okay, that's cool. What actually controls them? So we used a range of approaches, which I'm not going to go into, but essentially we found a master on switch that triggers all of these genes to be activated all at the same time. There's no point in activating 1 or 2 and making only some of the enzymes you need. You need to switch them all on at the same time. And Pseudomonas has got a crafty mechanism for doing exactly that. Okay, so this is it. Here are the genes just waiting to make their enzymes to make our magical chemical. So what Pseudomonas has is a gene encoding a particular protein that I've called activator because its job is to activate and activates these genes to make them work. When it does, then all the steps that I talked about before happen, and the enzymes get made, and the chemical gets made, and the iron gets taken up, and the bacteria smile and are happy. So this is different from the species that I talked about before, although it now turns out to be quite a common way of controlling production of these chemicals. So that told us a lot about making the iron plating compounds. What about the things that actually contribute to the infections, toxin and the protease? And it turns out that it affects those as well. So toxin and the protease, just like the iron plating compound, are controlled by genes. Okay, so here are our genes. And our activator can activate those as well. And what will happen is that the toxin, the protease get made, and that helps the bacteria to cause the infection. So this fitted with our original idea. We're really delighted this all fitted together because our original hypothesis that a single mechanism of controlling production of this chemical and also of these proteins had turned out to be right. So about, I don't know, 10 or 15 years after we started, it turned out that was true. That's cool, but the bacteria are making this stuff and making these things. They're going to be taking up iron, they hope, and eventually they're going to suffer from iron overload. How do they get around that? How do you switch it off? Okay, so essentially we have a similar kind of protein to the one I talked about earlier on, which is an inhibitor protein that's only active when the iron is present inside the bacterial cells. So what that does is it binds to the iron. When that happens, it joins on to the gene encoding the activator protein to block it, to stop it from working. And that is going to switch everything off. It'll stop the whole chain of events, and then, magically, nothing happens. So the activator protein goes away, then the genes that it would normally be activating just go silent again, the enzymes don't get made, and this compound is not going to get made, and everything goes quiet. Okay, so I think we're getting towards enough of those kind of circuits, but last point to make that is what we found subsequent to that is that the activity of the activator protein is controlled not only by how much iron is inside the cells, but by a second protein that basically joins onto it to stop it from working. So this protein joins onto the activator and then it can't do anything, even though it's present inside the cells. And a second switch that I'm not going to really talk about controls this guy so that he can be active or inactive. And if the regulator says go to it, then these genes will be expressed. The regulator says, no, we're going to turn things off, then these genes will be dormant. Okay, so where are we at? Really what we're doing now is to say, okay, we've identified the circuit inside the bacterial cells. We're looking hard at the mechanisms that control its activity. And the other thing that we're doing is really saying, okay, we've established this as one paradigm in Pseudomonas aeruginosa. If we look at other similar systems, do they work the same way? And the answer, rather surprising, is other systems that you'd think the genes say we're working the same way. When you actually go into the details, it's like, this isn't the same at all. So we're trying to figure out what are the similarities and differences between systems that actually appear to be just the same. Okay, so that's probably the most heavy-duty part of my talk. What I want to do is indulge myself for a couple of minutes and talk a little bit about this subject, how is pyroverdin made? So what I've done, we've been talking very happily about putting together bits of jigsaws and having balls of string doing the work, but in biochemical terms that's less defined than I'd really like. Okay, and that didn't quite satisfy me because the whole reason I got interested in biochemistry in the first place was I liked the idea of chemical reactions inside cells and I liked the idea of what's actually going on. So what I said was, look, I'm never going to be as famous as those guys in the 1940s and 50s that discovered all the great biochemical pathways that everybody knows and loves, although I do have the advantage that I'm not yet dead. So that gives me the opportunity to figure out a pathway of my own. So what we did was we identified basically all of the enzymes that are needed for making this compound, which is 15. We determined the functions of about 3 or 4 of them actually by chemically characterizing the enzyme in the test tube. The rest we can make very good predictions just by analyzing the sequences of those enzymes. And that allowed us to propose a pathway for making the pyroverdin compounds. I'm not going to walk through this, I just want to put up this diagram to show that it's quite complicated, but we were happy with it. In fact, we've done quite a lot of work since we proposed this originally that actually matches up with the functions we ascribed to the different enzymes. So I don't think I'm ever going to be in the center of the textbooks, but I can say, hey, look, at least we figured out one biochemical pathway. Okay, blank. So blank says, all right, we've had great fun doing all these kind of things, looking at the bacteria in the laboratory. We've learned a lot about how they work, figured out a lot of fundamental things. That's very nice, but the question is, how does that actually relate to disease? Okay, we can do all these experiments in the lab, but how does that actually connect in to real life, if you want to call it that, to what happens inside the patients? Pseudomonas infects people. So what happens inside the patients? So cystic fibrosis is one of the diseases that I talked about that Pseudomonas provides a problem for, and as Kurt said at the start, there's a real problem that the bacteria are very hard to eradicate. They're very resistant to antibiotics. Cystic fibrosis is a very common lethal genetic disease. About 500 patients in New Zealand. This kind of number in a country the size of New Zealand means that everybody in the audience is going to, well, many people in the audience will know somebody who knows somebody with cystic fibrosis. What happens is that the lungs of the patients become chronically infected with the bacteria, and this is bad news for the patients, okay? Because it's been shown that once they become infected, then their health declines. And what will happen with the patients is that they'll live, their life expectancy is greatly reduced. It's presently roughly about 40 years on average. Some patients will live a lot longer than that. Some will die a lot sooner. Okay, so here are pet Pseudomonas living in the lab, on a gel, in a soup, whichever. Oops. You don't need to be a scientist or a microbiologist or biochemist to tell that this is different from this. Okay, but here, Pseudomonas are living inside the lungs of a patient with cystic fibrosis. This is a very different place for them. So quite likely, their whole chemistry, their whole how they work is going to be very, very different from what we see in the lab. And that's fine, but how can we actually analyze that? So that's a problem that we started wrestling with a bit over 10 years ago. And we made a little bit of progress. And then what happened 6 years ago was that out of the blue I got an email from this guy, Dr. David Reid, who's a clinician, a respiratory physician who works in Hobart in Tasmania. And his job in Tasmania was to look after all of the patients, to manage the health of the patients in Tasmania who have cystic fibrosis. So he got in touch with me, as I say, out of the blue, but he was very interested in this question of how does iron fit in with what the bacteria are doing in cystic fibrosis. And I think he'd found some of our papers and figured we might be able to help him answer some of those questions. And that connection has really taken things a long way forward. So, cystic fibrosis, this is a picture that David provided. So this is actually an X-ray of a patient with cystic fibrosis, and this kind of clouding is telling you that the bottoms of their lungs they should be nice and clear. They're a bit blocked up with general gunge. This is some of the gunge that the patients cough up. So every month, then the patients go to a clinic to have a checkup. They get their general status checked, but they also provide a sample of all the stuff that's in their lungs for analysis. And this is the kind of thing that you get. Not very nice, and you can see why the patients aren't very well. What you can isolate from that, these are some Pseudomonas bacteria. As you can see, these look very different from the ones that I showed you before. What happens is that as the bacteria grow in cystic fibrosis, they change into a different kind of a form, this much more gel-like form, and that's not going to do the patients any good at all. This is really emphasising what I said before, that in the lungs of the patients, the bacteria can be behaving quite differently from when they've just been spending their entire lives living in the laboratory. Okay, so what do we decide to do? So we said, all right, instead of growing the bacteria in the lab, we're going to actually look at what they're doing inside the samples that come out of the patient. And as close as we can get to actually looking inside the patients themselves. So what we've done is to collect sputum samples from the patients, which David did for us, and then he sent those to us for analysis. What we want to do is to analyse the iron uptake pathways of the bacteria actually as it's happening in the sputum itself, which effectively is the same as in the patient. So how can we do that? Well, I mentioned already that pyroverdin is a beautiful fluorescent molecule, and as well as looking at it for many hours, just thinking how wonderful it is, we can take advantage of that property to actually detect it in complex mixtures. Fluorescent molecules have got very special spectral properties. That means that even if you've got lots of other things there, you can detect them very specifically. So that's basically what we did. We used this to measure the amount of pyroverdin in sputum samples. We applied to about 150 samples from 31 different patients. And what did we find out? We found out that in many samples from most of the patients, our compound pyroverdin was present. So that says bacteria don't do anything for nothing. If they're making it, they must be using it to be able to obtain iron to cause infections. But in some patients, we didn't find any pyroverdin at all. So that's a bit of a puzzle. And some patients were sort of in between. In some samples we'd see small amounts of pyroverdin, but in others we'd see none. So that was our first effort with that approach. The second approach we decided to use was to say, all right, instead of looking at the actual pyroverdin, we'll look at the genes that are being used to make it. Because if those genes are active, then that says the bacteria are making pyroverdin. If they're not active, then the pyroverdin is not being made, like I talked about before. So basically we developed a method for saying, are these genes active inside the sputum sample? Measure bacterial gene expression, I should have put activity in the sputum. Okay, so what did we find? So when we looked at the pyroverdin genes, we looked at 3 different genes with these particular names in a number of patients, and this is essentially the range of values that we got. So this said, in most of the patients, the genes are active. In other words, the pyroverdin is being made, although the amount varies between patients. And that's exactly the same conclusion that we got when we looked at the actual pyroverdin chemical itself. But in some, the amount is very low or negligible. And some, we couldn't detect any expression. So that says that our magical wonderful compound is very important, but not the whole answer to how the bacteria are actually obtaining iron inside the patient. We can make it crucial in the lab, but inside the patient, there's more going on. So we said, well, the bacteria, it's not like they've evolved some fantastic way of living without iron. they must have found some other way of getting hold of the stuff. So we looked at various examples. I'm just going to talk about one. It's probably the last thing I'll be talking about. What about other iron uptake systems? One example, uptake of heme. So heme is a chemical that's present in people in large amounts. We all know about it because that's what gives your blood the colour. It carries the oxygen around your blood. It's part of a protein, hemoglobin. So you can see some of that might be present in the lungs of the patients if their disease is pretty bad and if some blood was leaking into their lungs. Even just trace amounts would be enough to give the bacteria a bit of iron that they could use. So we developed the same kind of mechanism for looking at the presence of heme uptake systems, heme acquisition systems in the bacteria in the lungs of the patients. And these are different genes that we looked at to say, can the bacteria take up heme inside the patients? And the answer is that yes, they can, is the implication. In some cases, the expression of these genes is very high, and that says the bacteria are very actively expressing systems for acquiring heme inside the patients. And if they can do that, probably don't need pyoverdin, or perhaps that backs up the pyoverdin system. So that says these systems are active in cystic fibrosis. Okay, so that's giving us insights into what's actually happening inside the patients. Can we apply this at all? Well, as a start point, the kind of method that we've used for analysing samples inside the sputum, we can use that for looking not just at pyoverdin genes or heme genes, but lots of other different genes. So, in principle, with that approach, we can get a good idea of how the bacteria are actually behaving inside the patients where it really matters. There's two aspects of all the work that I described that we're exploring in terms of can we actually make people better. One, which is really initiated by David Reed, is to say, all right, bacteria really need iron if they want to be able to grow. And all our research is telling us that they're actively acquiring iron inside the patients. So in principle, if you can starve the bacteria of that iron, they'll be stuck. They won't be able to grow anymore. So can we add what are called iron chelators, iron-binding chemicals that will starve the bacteria of iron so they can't grow? So that's something we've been exploring a bit with David. Second approach that we've used is the bacteria, Pseudomonas bacteria, take up this compound pyoverdin very well. Now a big problem with Pseudomonas is that it's very resistant to antibiotics. And one reason is the antibiotics just can't get inside the cells. They're sort of bouncing around off the outside of the cells, but they can't get in where they need to actually be to work to kill the bacteria. So the concept is if you join antibiotics on to pyoverdin, the bacteria will take up this and inadvertently take up the antibiotic as well and then die. And in principle, it's been shown that can work. So we're doing some work on that with a colleague, Marv Miller, in the USA, who came here as a sabbatical visitor a couple of years back. Okay, so time to start wrapping up. So I don't want to do the Emmy thing and have thanks to everybody. So nonetheless, thank yous to lots of collaborators. So science these days is very much not one person beating away by themselves like it might have been 100 years ago. It really involves teams of people. And I really want to give a very sincere thank you to all the people that I've worked with. I thought the easiest way of listing all those was to write all the people that have been on papers that I've authored. So these are all of my co-authors. I think if you're not there and you think you should be, then let me know and we'll see what we can do about it. I've been hugely lucky over the years to have wonderful people working in my lab, right from pretty much when I started right up to right now. And this is listening to all those people. It's a real, one of the great privileges of being a university academic is that you get the chance to mentor, to train, to encourage, to support young people starting their careers in science. And I've been very lucky that way. So this is basically a list of the people who've worked in my lab, and it's been great fun, and hopefully they've got something out of it. I know some of them have. So thank you to all of them. Last of all, outside the lab, so Bowman Carlyle was very patient, tolerant and skilled in creating a lot of the figures that I used for this talk. The Department of Biochemistry, like I said earlier, has been a fantastic place to work. So I've had great colleagues. One very supportive head of the department has been really good. So I'd like to acknowledge them very much. These are the people that have provided the money, many of them external to the university. The university itself has been very good about supporting our research, and I think that funding has been really crucial as providing leverage for getting funding from external organisations. So I think the university manages that side of things very well. And lastly, probably most importantly, I'd like to thank my family for all the support, tolerance and understanding as I sort of work crazy hours and weekends and head out of town for a day or two or a week or two, and they all seem to welcome me back again. So, okay, and last of all, thank you very much for your time.
Iain, thank you very much for a wonderful talk. Inspirational, exciting, interesting, just extremely well done. It's been a great evening. I hope you guys have enjoyed it. It's been a great occasion to have your family here with you as you shared your work with us. It's been great to have colleagues from our own department. As you can see, we have a huge IPO crowd. It's been great to have colleagues from other departments that I can see throughout the audience. And I'm charged with giving the official response to your talk. I don't have much to say, but I've got 6 small points to say about things that I learned from this talk. And I say I absolutely love this talk. Some of them have to do with science, but many of them don't. One of them is I was just reminded about what a wonderful sense of humor that you have and what a warm and deep enthusiasm you have for science. And so it was very inspirational for me too, and very enjoyable for me. I was reminded again that you have really bad penmanship. And then I found out you have people in your lab that with great penmanship. I learned there's a song that has, it's not what you have, it's what you do. I've got to hear the song. So
give me a beer or two, I'll give you a rendition.
But I think the overarching theme I've seen is enthusiasm for understanding life at the molecular level. And not being willing to sort of give up on a problem until you have it solved. That's sort of my next point. And I think you've presented an impressive, a mature, a complete, and exhaustive body of work that is a deep contribution to the molecular basis of life. And that's an incredible thing to bring. And I think we're very grateful for that. And then further, that you've actually taken this next step and begun to address how this relates to patients in the clinics. So it truly spans sort of the bench to the bedside kind of thing that all of us would like to do and aspire to do. And you did this at a time when the tools were much more primitive. And the thing I sort of want to close on is that now's this time when we have deep sequencing and we can sequence entire organisms and we can do these powerful genetic experiments and we can do structural biology and we can do the entire proteome. So the sorts of questions that you're addressing now are even bigger and wider and even more impact. And so I think with reference to Professor Lamont, that I'll quote the words of another song, you ain't seen nothing yet. Thank you very much.
And so it just leaves me to give Iain a small token to say thank you for the lecture and congratulations again on your promotion.
Thank you very much.